Hello everyone, so this video is going to be covering red blood cell metabolism. Now, the sort of ironic thing about red blood cell metabolism is that red blood cells are actually anaerobic, and that means that they don't require oxygen for their own metabolism, even though that red blood cells are responsible for carrying oxygen to all the other parts of the body. The main key takeaways from the fact that red blood cells are anaerobic is that we get our ATP from glycolysis, and glycolysis alone, and also that we get NADPH from the pentose phosphate pathway. Now, why are these two things important? Well, when we're making ATP from glycolysis, that means we're going to have a bunch of 2,3-BPG. And we know from the other lectures already that BPG is going to decrease hemoglobin's affinity for oxygen, so it's going to promote O2 release. But it's worth pointing out that in states of hypoxia, when we have low O2, we're actually going to induce more BPG to be synthesized. Now, what about the pentose phosphate pathway? Well, NADPH, it turns out, is an extremely important molecule for regenerating glutathione. And glutathione is a very powerful antioxidant. So glutathione is responsible for detoxifying hydrogen peroxide. So when glutathione hasn't been regenerated, it actually forms a disulfide complex with another glutathione molecule. So I'll just abbreviate that as G. And using an enzyme known as glutathione reductase, we're going to disassociate those two glutathione from one another. So we just get two GSHs. So glutathione with a sulfide group. And we do this specifically by utilizing NADPH. And it will be taken down to NADP minus. Now, to then detoxify hydrogen peroxide, we simply take our glutathiones and we're going to re-put them into the dual glutathione disulfide bridge complex on one side, and then by doing this we are able to take hydrogen peroxide and detoxify it just to water. So why do we care so much about detoxifying hydrogen peroxide? Well, it turns out that in cases of oxidative stress, when there are a lot of oxidants in our blood, that the iron atom within our hemoglobin is going to become oxidized. It'll go from an iron 2 plus to an iron 3 plus. And in the 3 plus state, it's unable to bind with other oxygens, meaning that we've just rendered our hemoglobin useless, because the whole point of having hemoglobin in our red blood cells is to carry oxygen around. So if in cases of oxidative stress, we're no longer able to transport oxygen. So a decrease in oxygen carrying capacity, which means that we're going to get cyanosis, which is a hypoxic state. So we'll turn blue. So now, what causes oxidative stress? Well, it turns out we're always encountering oxidants in our environment. They're everywhere. We aren't really able to reduce the amount of oxygens we come into on a day-to-day basis. What we can do, however, is we can increase our antioxidant capacity. So we can increase our ability to detoxify these oxidants as we come across them. But what happens in cases of oxidative stress is that we lose some capacity to produce an antioxidant. So in the case of our pentose phosphate pathway, we're going to be producing NADPH. That's the point of the pentose phosphate pathway. One of the enzymes to do this is going to be glucose 6-phosphate dehydrogenase. But if we are deficient in glucose 6-phosphate dehydrogenase, that means we're not going to be able to make enough NADPH to regenerate our glutathione. So in cases when our glucose 6-P dehydrogenase isn't producing enough NADPH, we would call this a G6PD deficiency. This would cause oxidative stress and we would get cyanosis. So next I want to talk about red blood cell membrane structure. And this is important because the membrane structure is dependent upon the membrane proteins that make up that structure. So the proteins themselves can be either peripheral or integral. And I have this drawing here. So a peripheral protein would be any of these ones here that are sitting on the periphery of the membrane, whereas an integral protein are going to be any of the proteins that are within the lipid bilayer. So this band 3 would be an integral protein because it's spanning the length of the lipid bilayer. So these different types of membrane proteins are going to be determining that biconcave structure of the red blood cell, as well as providing anchor points like this anchorin here for cytoskeletal elements to provide more structure and stability. Now, proteins themselves are extremely difficult to isolate. So all of these proteins, because they are within the lipid bilayer membrane, they have to be boiled in a detergent known as SDS. And that, by boiling in SDS, we will solubilize the lipids in the lipid bilayer membrane, as well as reducing disulfide bonds, which add to the structure of and the integrity of the membrane with the proteins. So what happens if we have a problem with these proteins? We have some sort of mutations that's affecting them. Well, we can have a condition known as hereditary spherocytosis. In hereditary spherocytosis, we're going to have a mutation in either spectrin, ankyrin, or band 3. And this is what causes a loss of a vertical interaction. So a vertical interaction would be any of these elements that are promoting the vertical stability, so up and down, of the membrane. Whereas in a condition known as hereditary liptocytosis, this is going to be also cause a problem in spectrin, but instead of band 3 or ankyrin, we're going to have protein 4,1, which is a reference to the band number of that protein as well. So like band 3, protein 4,1 is just another band number, and glycophorin C. This is going to be causing an interruption in the horizontal component of the membrane. So what happens if we have one of these conditions? Well, it turns out in capillaries, we can only get one red blood cell through at one time. So if we have an improper structure, we can't push those blood cells through as easily, meaning that we'll end up with anemia because we can't transport our oxygen as well as we could. And we can also get jaundice because our spleen will pick up the fact that we have improperly shaped red blood cells and will try and filter them out, but they'll filter so many of the red blood cells out that the metabolic products from doing so will build up in our system. Now finally, I want to talk about ABO blood groups. You probably already know there's 3 different main types of blood groups. You might even know your own. Each of the different groups stand for different oligosaccharides that actually stick out on the red blood cell surface. So you can see I have 4 different red blood cells here, and this is a type O. A type O is going to have what we call a terminal fucose group. And I've just represented that by these little white sticks. So for all of these blood groups, they all have this terminal fucose, as you can see here I've drawn it in. So it will always have the terminal fucose, which I'll just abbreviate as TF. So type A will have a terminal fucose, type B will have a terminal fucose. But the difference being that type A also has what we call an N-acetogalactosamine. And this ends up being a side chain, like you can see here I drew in with these blue dots. And finally, for type B, we have our terminal fucose again, but we also have this time just a simple galactose side chain. And so a type AB is just going to have a mix of both the N-acetogalactosamine and the galactose side chains. Now these side chains are going to end up to be acting as antigens for our immune system. So our immune system will recognize our own antigen. So say like I myself am a type A blood group. So I have this N-acetogalactosamine on my blood, but if I received type B blood, I would produce antibodies against the galactose side chain. I would have an immune response against that type. And so type O then would have an immune response against all of the different types of blood except for itself, but it can give to any type it wants because all the types have that terminal fucose. So type O is known as the universal donor, whereas type AB is going to have both side groups, therefore it won't produce any sort of immune response, but at the same time it can only give to type AB. So it is known as the universal recipient.